Hello guys, Jai Shri Ram. Today we are going to see heredity and evolution. It is chapter number 1 in science 2 of SSC of the SSC board. Okay. It has 40 slides total. So the lecture could be almost of 1 hour or so. Uh, in the video description, you will find this material, that means this PPT, as well as index. So whichever part you find interesting or you want to know more about, just click the timestamp ahead of that particular part and the video will jump to the time when I have explained that particular part. Okay, so let us start with this hour-long video. First things first, let me tell you the basics. Genes determine our hereditary traits using proteins. Okay, so let me explain this. Okay, so all the observable characteristics of any organism are called as the traits of that organism. Heredity, also called as inheritance, is the transfer of traits from one generation to another using genes. Okay, gene is considered to be a basic unit of heredity or inheritance. See, any body, whether of a human being or a plant or an animal, any body basically consists of organs. Organs are made up of tissues. Tissues are made up of cells. So cell is the basic unit of any body. Okay. Now, every cell is made up of three parts: nucleus, cell membrane, and cytoplasm containing cytoplasmic organelles. Cytoplasm is a jelly-like fluid which fills the space between nucleus and the cell membrane, and there are so many small organelles floating around in that jelly-like liquid, and they are called as cytoplasmic because they belong to the cytoplasm. They are floating inside cytoplasm. And organelles because they are not organs, they are not well developed organs, but they have some specific use in the cell. And so organ is what? Organ is also some part of the body which has some specific use. So organelles also have some specific use, but not in the body, in that particular cell, and that is why they are called as organelles. So cytoplasmic organelles. Nucleus. Now, out of all these three things, we'll only focus on the nucleus at the moment. Nucleus has two vital nucleic acids. One is DNA, another is RNA. Nucleus has 23 pairs of chromosomes. I'll explain more about it. DNA is not always in the form of chromosomes. Chromosomes is the shrunk and well packaged form of DNA. Normally, it is just floating around in the nucleus. So I'll tell that in the next slides. Chromosomes are neatly packaged DNA molecule. DNA is a single molecule. So look at how small these things are. Look at the scale of things. Cell, you can't even see a cell. Inside that there is nucleus. Inside that there is DNA. DNA is just one molecule. Can you see a molecule? No, molecules are very small. DNA is one single molecule. One chromosome means one DNA. Gene is a segment of that DNA which is responsible for a certain trait. That means for one observable characteristic, you will have one gene on the DNA. So DNA, one DNA molecule can have many genes, and every gene will be a series of nucleotides. That will also come in the next slides. Okay, all the organisms except viruses have DNA as their genetic material. This is also an important thing, maybe asked. It's coming directly from the textbook, and yeah, I think this is very important for you to know. Even if you're giving NEET exam in the future, this will be very important, that every organism on the planet except viruses have DNA as their genetic material. And that is why viruses require some host to proliferate. Proliferate means grow in numbers, reproduce. Okay. Now, how is a cell? Cell is something like this. Okay, you can go through it. I'll explain in the next slide. Cells are of two types. Prokaryotic cells means unicellular organisms are basically a single cell, right? And those are called as prokaryotic cells. So unicellular organisms without nucleus and cell membrane, membrane enclosed organelles. So they will only have cell membrane, they will only have cytoplasm. Inside that, everything is khichdi. Everything is mixed up. They don't have a nucleus, they don't have a mitochondrion, they don't have different organelles. Everything is mixed up within that jelly like fluid called as cytoplasm. Okay, so those are called as prokaryotic cells. They don't have membrane enclosed organelles. Organelles are not very well defined and they are not covered by a membrane. Okay, but cell membrane is there. Every cell has a cell membrane. Cell membrane is always there. Eukaryotic cells are the cells of the advanced organisms like us, other plants or animals. Okay, advanced animals, they use, or multicellular organisms, they usually have eukaryotic cells. So here, how is a eukaryotic cell? It has a cell membrane. Of course, every cell has a cell membrane. Even prokaryotic cell also has a cell membrane. Then it has a nucleus and it has cytoplasm with cytoplasmic organelles. Now, cytoplasmic organelles are endoplasmic reticulum, ribosomes, mitochondria, Golgi bodies, and lysosomes. So, we will see all of them in the next slide. Cytoplasmic organelles are specialized structures within the cell that perform specific functions. Some of the common cytoplasmic organelles are mitochondria. It is responsible for generation of energy within the cell. So, these are the powerhouses of the cell. They do it using cellular respiration. Endoplasmic reticulum, it is involved in protein synthesis and transport. Endoplasmic reticulum can be either smooth or it can be rough. It's the homework for you to find out what is the difference between the two. Let me know in the comment section. Ribosomes, they are responsible for protein synthesis. Liposomes are containing some digestive enzymes that break down and recycle cellular waste and foreign substances. So what it means is, let us say, I have to break down certain things. They are no longer required. Okay, so they will be consumed by lysosomes. Mitochondria will actually consume good nutrients and they will generate energy. Okay, so food that you eat, that will be consumed by mitochondria. Oxygen that you breathe, that will be consumed by mitochondria for cellular respiration. But all those kind of things that you no longer require, dead kind of things which just need to be discarded, so they get digested or discarded in a way using lysosomes. Golgi apparatus is involved in the protein modification and packaging. So what happens is, inside the nucleus, ribosomes, rRNA gets created. rRNA is nothing but ribosomes. Okay, these ribosomes get settled down on endoplasmic reticulum. Okay, so they sit on the endoplasmic reticulum. Okay, they do the protein synthesis, so protein will get created by that. Okay, then this protein travels from endoplasmic reticulum to Golgi apparatus, also called as Golgi body. So when it goes there, then this protein is also one molecule. Okay, one molecule, it is called as polypeptide. So it is basically many amino acids connected by peptide bonds. So many peptide bonds, that's why polypeptide. Protein means polypeptide. Polypeptide is another name of protein. So it is one molecule. Now this molecule is then attached with carbohydrates or with lipids in Golgi apparatus. Okay, then maybe they also undergo some structural changes. See, a protein, actually how your body looks or behaves, means the structure and function of your body is determined using proteins. But proteins cannot do it just like that. They have to be modified a bit. They have to be put in a certain structure. Then only they are able to do those functions, and that putting them into structure is done by Golgi apparatus. Also adding them with carbohydrates, adding them with lipids so that they can do the bodily functions, that is done in the Golgi apparatus. So Golgi apparatus is also very important. Many people don't treat it with as much respect as you should be treating Golgi apparatus. Okay, so this was all revision so far. Nothing of this, whatever we discussed, none of that is going to be part of this lecture, but you will require this knowledge to understand this chapter. Now the chapter starts. Let us start with DNA. So cell's DNA is something like this. It is very, very long molecule. It's a single molecule, very, very long. Only during reproduction it is packaged nicely, and this is how it looks. okay, when it is packaged nicely, and that is called as one chromosome. So DNA exists in the nucleus. Yeah, DNA always exists only in the nucleus. It never comes out of the nucleus. Okay, in case of eukaryotic cells. In prokaryotic cells, everything is floating around, even DNA is floating around. But in eukaryotic cells, DNA always exists in the nucleus. It never leaves the nucleus. Okay, RNA is generated in the nucleus. It leaves the nucleus, comes in the cytoplasm. That happens. RNA floats around everywhere, but DNA always stays within the nucleus. Now DNA exists in the nucleus. It is in tangled form called as chromatin. So normally when the cell is in a normal state, it is always floating around. Okay, it is always in the open state. So such a long molecule, you can imagine that it must be tangled everywhere. Okay, and how many DNAs do we have? We have 46 DNAs in a single cell. Okay, so all of them are just floating around in the nucleus, and that thing is called as chromatin. Okay, only during reproduction DNA is packaged nicely into one structure which is called as chromosome. Okay, there are 23 pairs of chromosomes in every single cell of a human being. Okay, so total how many chromosomes? 46. Means one chromosome is one DNA molecule. So how many DNA molecules do we have in one cell? 46 DNA molecules. 22 chromosomes are called as autosomes and 2 are sex chromosomes. Okay, now human body has 46 DNA molecules. They are together called as human genome. So how will a human being generate? See, from a single cell also I can generate the entire human body. How does every cell know how the human body should be? Every cell has these 46 DNA molecules, and these 46 DNA molecules together is called as that human's genome. Okay, I can generate Amit Shirasao from any single cell of Amit Shirasao's body. Okay, that is called as my genome. Okay, now I'll explain you. This is the most important slide. What are chromosomes? What is DNA? What are genes, codons, amino acids, and proteins? So basically, chromosomes lead to the creation of proteins through all these stages. So I'll explain them one by one. Each cell has 23 pairs of chromosomes, right? Each chromosome has one neatly packed DNA molecule. Okay, means basically chromosome itself is each chromosome is wrong. Each chromosome is a neatly packed DNA molecule. This is correct. Okay, each DNA has several genes. Each gene is made up of several nucleotides in a sequence. What is a nucleotide? It will come in the next slides. But just remember that each gene is a series of nucleotides. How many nucleotides? 700 nucleotides, 600 nucleotides, 1000 nucleotides, something of that order. I'm not talking about 20, 25, not like that. 700 nucleotides in a sequence. That will together be called as a gene. So imagine how long that DNA must be. Codon means triplet, also called as triplet codon, is a group of 3 nucleotides. So 3 consecutive nucleotides are called as codon. Why only 3? Why not 2? Why not 4? Okay, because our RNA reads nucleotides in a combination of 3. It reads 3 nucleotides at a time. Every codon represents a specific amino acid. Okay, and that is why our RNA reads one codon at a time, so that it can decipher which amino acid I want to use for the protein synthesis. Okay, so what is a protein? Protein is nothing but a polypeptide. Means what? There are many peptide bonds between amino acids. So one amino acid molecule, peptide bond, another amino acid molecule, peptide bond, another amino acid molecule, and that's why there are so many peptide bonds that a protein is called as polypeptide. Poly means many, peptide means peptide bond.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins.
Okay, I hope you got it. So basically, the link to this video is also given in the last slide. You can go through this video. This is a very good video. It will give you a visual description of how a human cell is. So here, basically what we saw is how is DNA and how are genes on that DNA. Okay, let's go ahead. What is DNA? DNA is deoxyribonucleic acid. So basically the way to remember it is deoxyribonucleic acid. Why? Because RNA is ribonucleic acid. So just remember ribonucleic acid. Ribo means ribosome. So it is see, RNA is created in the nucleus, and the rRNA is basically the ribosome. Okay, so ribonucleic acid, it is an acid. As I told you, there are two vital acids in the nucleus, DNA and RNA, right? So it's an acid. So nucleic acid, and RNA means rRNA is basically ribosome only. So ribonucleic acid, that is rRNA, and deoxyribonucleic acid is basically DNA. Okay, so remember ribonucleic acid and then just add deoxy in front of it to make DNA. Got the idea? Now, how is a DNA structure? Let us see. Watson and Crick, they gave us a double helix DNA structure. So they said that DNA is like a ladder. Okay, there are two poles in the ladder, and then there are small steps of the ladder. Okay, now this ladder is twisted, means the two poles are causing helix. So I mean, helix means what? One pole curved around like this, that is called as a helix. Now there are two such helix near to each other, and they are connected by this kind of steps. So that is why it is called as a double helix structure. You can remember it like a twisted ladder. Okay, that they should be calling it twisted ladder structure. I don't know why don't they use simple words, but they call it double helix structure. DNA is made up of three main things: pentose sugar, phosphate, and 4 nitrogen bases. Okay, so what are the 4 nitrogen bases? Adenine, guanine, cytosine, and thymine. So this is how it looks. Basically this is a pentose sugar. Pentose because it is basically a sugar molecule which has the shape of a pentagram. Pentagram means polygon having 5 sides, just like you can say Pentagon of the US. It's a pentagram. So this sugar molecule is in the shape of a pentagram, and that's why it is called as pentose sugar. Okay, and then pentose sugar along with this phosphate, they together form this kind of a helix. So one helix is made up of pentose sugar and phosphate connected in a series. Okay, now this is also called as the backbone of DNA, also called as the one helix of DNA. Okay, the two backbones are then connected using nitrogen bases. So here pentose sugar is connected to a nitrogen base called as adenine. Here it is connected by guanine, cytosine, thymine. Okay, now adenine will always connect with thymine using 2 hydrogen bonds. Guanine will always connect with cytosine with 3 hydrogen bonds. Here again it is cytosine, so it will connect with guanine. Here it is thymine, so it will connect with adenine. Okay, and this is how the DNA stays in place. I mean, both the two helix stay in place due to these nitrogen bases connected by hydrogen bonds. Got the idea? This is the DNA structure. A and T are always joined by 2 hydrogen bonds, and G and C are always joined by 3 hydrogen bonds. This can also be an interview question. I don't know why it's not asked, but at least if you are preparing for NEET, then this is an important question. Now, what is a nucleoside and a nucleotide? Nucleoside means this pentose sugar along with the nitrogen base, they are together called as a nucleoside. Okay, nucleoside. Now, if you combine this phosphate group also into it, then it is called as a nucleotide. Okay, so nucleoside, nucleotide. And then I told you that many nucleotides, a series of nucleotides is called as a gene, and a series of genes is called as a DNA. Got the idea? So DNA is made up of many nucleotides. Okay, so jellyfish emits a green fluorescent light. Okay, and that's why it is called as bioluminescent. The property is called as bioluminescence. The trait is called as bioluminescence. Now it is one trait, so one trait must be due to one or more genes. Let us say there is one gene responsible for this trait. How long is that gene? How many nucleotides? It has these many nucleotides. It has 700 nucleotides in a certain sequence. Then it is called as a gene. Okay, so let's go ahead. What is an RNA? RNA is ribonucleic acid. I already told you. Robert Hawley told us that it has a single helix structure. It is just like a one leg of ladder, and it is also twisted. That is the single helix structure suggested by Robert Hawley. RNA is made up of 3 main parts again: pentose sugar, phosphate, and 4 nitrogen bases. So till now everything is common, but these 4 nitrogen bases are adenine, guanine, cytosine, and uracil. Instead of thymine, there is uracil in RNA. Okay, so there are two basic differences between DNA and RNA. First thing is DNA is a double helix structure, RNA is a single helix structure. Second difference is instead of thymine nitrogen base, RNA has uracil nitrogen base. And there is third difference also, that DNA always stays in the nucleus, RNA gets generated in the nucleus but keeps floating around. Okay, so you can find it in the cytoplasm as well, so you can find it outside the nucleus. So these are the 3 differences between DNA and RNA. Now RNA, let us dive deeper into it. There are 3 types of RNAs. Now these are the images that you will find in your textbook, very simplified images. In the next slide I'll tell you how they really look. So here single helix structure is there only for mRNA. tRNA looks like a sphere, maybe having one big sphere and one small sphere connected to each other. It looks something like that. rRNA, that is ribosomal RNA. Ribosomal RNA is nothing but your ribosome. Okay, rRNA is nothing but the ribosome. Okay, messenger RNA is mRNA. Then tRNA looks like a clover leaf. Okay, clover leaf model. 1, 2, 3. Okay, three globes, you can say. That is called as a clover leaf model. And then it also has this thing here, an enzyme. Here a amino acid gets connected. Okay, so this part here, the amino acid gets connected, and this part is the place where anticodon is there. I'll tell you in the next slide. But this is the diagram that is given in your textbook, very rudimentary or basic diagram, but I'll tell you in the next slide how they really look. Now let us go with protein synthesis. How is DNA, mRNA, tRNA, rRNA are involved for protein synthesis? DNA has genes. Okay, each gene is an information for protein synthesis, and proteins only determine our traits. Traits means observable characteristics of a person, okay, or of any organism. So our observable characteristics are determined by proteins, and which protein will be created, that is determined by genes, and genes are nothing but sequence of nucleotides on a DNA. Got the idea? This is how the whole thing is. Now mRNA carries the protein synthesis information from a gene of DNA to the ribosomes of the cell. Got the idea? So basically, genes are present on DNA, and DNA never leaves the nucleus. But then protein synthesis happens in ribosomes. Ribosomes are in the outside the nucleus, right, in cytoplasm. So who will communicate the DNA's information to ribosome? That is done by mRNA. Okay, so ribosomes are cytoplasmic organelle which perform protein synthesis in a cell. Ribosomes are also called as protein factories of the cell. Okay, now tRNA carries the amino acids to the ribosomes. Now ribosome has got the message that okay, this is how I have to generate the protein. But what is protein? Protein is a sequence of amino acids connected by peptide bonds, and hence it is called as polypeptide, right? So now ribosome has to create a polypeptide. For that it requires amino acids. Now where will it get it from? Now amino acids are also floating around in the cytoplasm. Who will bring those proper amino acids to this ribosome? Who will do that? tRNA does that. Okay, so amino acids are the building blocks of any protein. In a cell, amino acids are floating around in the cytoplasm. Now, structure of a tRNA, as given by Byju's, by the way, is like this clover leaf model. Okay, but this clover leaf model, it does not look so beautiful, really. It is tangled. This clover leaf is tangled around itself, and it actually looks something like this. Got the idea? Now here means this green part is this green part, this yellow part is this yellow part, this violet part is this violet part, blue part, dark blue part, dark blue. Got the idea? This is how it is. And what is this black thing? Black thing is the amino acid that is connected to it. So amino acid gets connected over here to 3' part. Okay, and anticodons exist over here. This is called as an anticodon arm. Okay, so over here, basically it connects to the mRNA using this anticodon arm, and it has the amino acid on this acceptor stem. Got the idea? This is how a tRNA is there. Okay, so tRNA carries this amino acid from the cytoplasm to the ribosome. Okay, now what happens in the ribosome? It basically connects using these anticodons with the codons on the mRNA, and then something happens. I'll tell you later on. Why should I tell you in this slide? I'll tell you in the next slide. rRNA. What is rRNA? rRNA is the primary component of ribosome. Basically, for simplicity, you can assume that rRNA is nothing but ribosome. Okay, but technically speaking, whenever you write in the exam, you can say that rRNA is the primary component of ribosome. Okay, just the way of saying it, but basically for simplicity you can remember both of them are exactly the same. It does protein synthesis using amino acids as per the message given on the mRNA. Got the idea? Okay, now how is an rRNA? rRNA is something like this. So it has this bigger sphere and this smaller sphere. So this is called as large subunit, and this is called as small subunit of the ribosome. Okay, or ribosome means rRNA. Okay, now here reading of the mRNA is a string like this. So reading of this mRNA happens from 5 prime to 3 prime. So that means this ribosome basically keeps moving across the mRNA. So it will be moving from here till here. Got the idea? This is how ribosome will be moving, or rRNA will be moving across your mRNA. Got the idea? Okay. Now ribosome has 3 sites. APE. Remember it like ape. Planet of the Apes, ape. Remember it as ape. See, if the ribosome is moving from this direction to this direction and it is reading from here to here, then definitely the new thing that it will be reading, the new codon, will be coming from this side, right? So the new codon will be received at the A site. So A site is towards the 3 prime side. Okay, So it will first, this is called as amino acid site, because the amino acid will get joined over here. I'll tell that in the future also. This is peptide side, that means then whichever, then it goes ahead, then the second amino acid gets connected. Now these two amino acids are connected using peptide bonds. So that bonding happens at the peptidyl site, right? Between these two sites it will happen, right? So peptidyl site is that. And then exit site means from here the tRNA which was carrying that amino acid, tRNA will dissociate and it will go away. And this is the long chain of amino acids connected to each other using peptide bond. This is nothing but a polypeptide. This is nothing but a protein. Got the idea? This is how the protein is synthesized. Okay. So that was a difficult diagram. In exam, you can draw this diagram. This is easy enough. So basically, ribosome is moving from 5 prime to 3 prime. Okay. So it is going from left to right. Okay. So the new codons will be read from this side, right? From the 3' side. And that is why this site is called as A, that is amino acyl site. Then here peptidal side, what is it called? Peptidal side, and this is exit side. So this is how this is the diagram that you can draw. That's the new one comes and sits over here. This is the tRNA, it is carrying an amino acid, so it comes and sits over here. Then the next time when it moves, then this thing comes over here, and this thing comes over here. It releases itself, and here peptidyl bond is created between whichever is the amino acid earlier, and then this amino acid. Okay, so this is the whole process. We'll see that in the next slides as well. In the exam, you can only remember this particular diagram, that is more than sufficient, and this is the slide that is more than sufficient for you. Okay. Now, see this visualization. It will clear all your understanding about how protein synthesis happens.
These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So this is the whole story of protein synthesis. Now let us see that word by word. We will analyze this, we will write it down. Okay. Now, there are 3 steps in protein synthesis. This can be an interview question as well, and all these 3 are very confusing. They have used T everywhere. Transcription, translation, translocation. They should really find better words, but it's really confusing. So this can be a good interview question, because people like to ask you where you get confused. So Transcription, especially the NEET students will require this. Transcription takes place in the nucleus, whereas translation and translocation will take place in the cytoplasm. So transcription means what? Enzyme RNA polymerase. In that video you saw, no? It all started with RNA polymerase. So when a gene is activated, RNA polymerase comes at the engulfs the DNA in a way, and it basically tears the DNA apart. Okay, and then it creates mRNA using free nitrogen bases which are floating around in the cytoplasm. Okay, so this is done. That is called as, this process is called as transcription. So transcription means reading one strand of DNA and creating a corresponding strand of mRNA. That is called as transcription. Okay, so it is shown in simple diagram over here. Okay, so this is that RNA polymerase enzyme, and this is that DNA strand. It is broken into two parts, and with any one part, it is now creating an mRNA. Where are all these nitrogen bases coming from? They are already floating around in the cytoplasm. They keep coming there based on whichever nitrogen bases are there on the DNA strand. Opposite nitrogen base will come and get collected. Okay, and they form one strand, and that strand is called as mRNA. This process is called as transcription. Translation, again, it takes place in the cytoplasm. Translocation also takes place in the cytoplasm. Translation and translocation go hand in hand. Okay, so I am giving the description in this particular image. Okay, you see very closely. What translation means is, as per the codons on the mRNA, so now this mRNA leaves the nucleus using the nuclear pore, through the nuclear pore, any one nuclear pore, and it comes into the cytoplasm. Now in cytoplasm, it binds with the small subunit of rRNA, that is ribosome. Okay, that small portion will move from 5 prime to 3 prime direction, and as soon as it gets start codon, then that is where the large unit also connects, and the initiation complex gets created. And then this initiation complex keeps on moving from 5 prime to 3 prime while generating the protein. And whenever stop codon, any one stop codon is encountered, there are 3 stop codons in human body. So if any one of them is encountered, then the protein synthesis stops. Again, the ribosome split. Everyone goes back to their own business. That's how protein synthesis happens. So now translation means what? As per the codons on the mRNA, the amino acids are brought by the tRNA from the cytoplasm to the ribosome. Okay, and every tRNA has anticodons, and that's why it is possible. So these anticodons match with the codons of the mRNA. So if mRNA has UGG, then U always connects with A, G always connects with C, G always connects with C. So the anticodon that can connect over here is ACC. So only that tRNA will come which has this anticodon, and that tRNA will obviously have a certain amino acid attached to it, right? So this is how the protein synthesis is done. So everything is happening as per the mRNA only. As per the code given on the mRNA only, these amino acids are connected one after the other, and this is how that protein synthesis happens. Now translation means basically creation of this amino acid chain. Creation of the polypeptide means creation of the protein. And translocation means this ribosome is moving one codon at a time from 5 prime to 3 prime. That movement is called as translocation. So you can remember by the word location. So it changes the location, translocation. And translation means basically you are translating what is given on mRNA into an actual protein, right? So text is being translated into actual protein. Okay, or codons are translated into actual protein, and that's why it is translation. Transcription you can remember, because transcription means you're writing some script. That's why transcription. So writing a script means you're reading the DNA, you're writing the script on mRNA. Okay, So you're copying the codons of DNA onto the codons of mRNA. So this is copying, writing the script, right? That's why it's called as transcription. So this is how you should remember it. Transcription is about scripting. Such a tongue twister, yeah. I mean, they should really use simpler words. Translation, because it translates the language of codons on the mRNA into actual protein. And translocation means it actually changes its location. Ribosome keeps moving from 5 prime to 3 prime across the mRNA. This is called as translocation. So this is the difference between all three of them. As a result of transcription, translation, and translocation, we get a long chain of amino acids connected by peptide bonds. This is nothing but protein. Hence, proteins are also called as polypeptide. I have repeated this 3 times now. At least now you should remember it, that proteins are called polypeptides. Okay. Now, protein synthesis transcription is explained in further detail. Do you think this is detailed, or now we'll go in further detail? Transcription is the process of formation of mRNA using DNA as the template. It happens in the nucleus. Detailed steps. Gene has promoter section and terminator section. Okay. Enzyme RNA polymerase binds to the promoter section and starts moving towards the terminator section. Obviously, in between promoter and terminator, there could be one or more genes, but from promoter to terminator only this reading will happen. Got the idea? From promoter to terminator only this mRNA will get generated, and accordingly proteins will be synthesized. Got the idea? So promoter, terminator. RNA polymerase, you already know about it. DNA with the RNA polymerase is unwinded and the 2 strands are divided. Yeah, we have seen this. The mRNA is formed from using one DNA strand as the template. We have seen this. Nucleotides from cytoplasm enter the RNA polymerase from the cytoplasm to make this happen. So how will this mRNA form? You require nitrogen bases, right? So those nitrogen bases will flow from the cytoplasm into the nucleus, and that's how you get it. Okay. mRNA is made up of several exons and introns. What is exon? Exon means exon includes the code for protein synthesis, and introns don't include information about protein synthesis. You saw in that video also, right, that mRNA has to be cut and only those parts are joined which have some protein synthesis information. Okay, this process, so the non-coding introns are removed. Also 5 prime cap and 3 prime tail are added. Okay, so this cap and tail are only to tell rRNA that, see, you move from 5 prime cap to 3 cap tail, 3 prime tail. Got the idea? That's why from 5 prime to 3 prime, rRNA has to move in that direction. That's why this cap and tail are attached on the mRNA. So this whole process of removing introns, adding the cap and tail, this is together called as intron splicing. Who does it? It is done by spliceosome. What is a spliceosome? Spliceosome is a complex of proteins and RNA. Proteins and RNA, they form a complex called as spliceosome, and it does this basically intron splicing. The mature mRNA exits the nucleus into the cytoplasm from one of the nuclear pores. Got the idea? So whatever exits from the nuclear pore, that is always a proper mRNA. Splicing is already done. Intron splicing is already done over it. So where does intron splicing take place? Inside the nucleus. All the steps that I have mentioned so far, all of them happen till here. Everything happens inside the nucleus. And in the last step, I have said that this mature mRNA exits the nucleus using one of the nuclear pores. Got the idea? Protein synthesis, initiation, elongation, termination stages. Now this is particularly for NEET students. This is not for 10th standard students, but still, it's good if you understand it. Okay. Protein synthesis happens in 3 stages: initiation stage, elongation stage, termination stage. Initiation stage means a ribosome is made up of two subunits, smaller and another large. The smaller subunit binds with the mRNA. Got the idea? First smaller one will bind with the mRNA. It moves along the mRNA from 5 prime to 3 prime until it finds the AUG codon, also called as a start codon or initiation codon. Okay, so there is one start codon, only one start out of 20 codons. 20 amino acids means 20 codons, right? So 20 out of 20 codons, this is the only codon which will start the protein synthesis, and that is why it is called as a start codon or initiation codon. So whenever that is encountered by the small section of rRNA, now the tRNA with activated amino acid is attached to it, binds due to the anticodon, and then the large subunit also binds. So now the rRNA gets complete. Small rRNA was anyways moving across the mRNA from 5' to 3' direction. It found start codon. As soon as it finds it, tRNA carrying a particular amino acid for that start codon, it will get attached over there, and on top of it, the large section of the rRNA will also get attached. Now the rRNA is complete, and this whole thing is called as the initiation complex. Large subunit has A site, P site, and exit site. I've already discussed them. The acceptor site, some people also call it as acceptor site, but I feel it should be amino acyl site, amino something. Let me check. It should be amino acyl site. Okay, so but many people have given acceptor site as well. So I will just mention for your ease. I'll mention. Okay, both these are correct names for that site. Okay, and the whole unit means two parts of the ribosome, mRNA and tRNA. This whole thing means these three things together. Ribosome means rRNA, messenger mRNA, everything is happening on mRNA only, and tRNA for transport. So both of all three of them, when all three RNAs come together, then this is called as an initiation complex. Got the idea? Initiation complex is this thing. Elongation stage, the initiation complex keeps moving from 5 prime to 3 prime, okay, across the mRNA, one codon at a time. So this is what translocation. Okay, so this translocation is happening, and due to this translocation, whenever you read a new codon, a new amino acid gets joined to the existing chain of amino acids using peptide bond, and this polypeptide is getting generated, means this protein is getting generated. So this is what, this is your translation. Okay, so translation and translocation together, they are responsible for elongation. That means the chain of amino acid is elongating. That means the protein is getting generated. Peptide bonds are created across the amino acids. Translation is the formation of the amino acid chain, and translocation is the movement of the ribosome, that is rRNA, across the mRNA from 5' to 3' or 5 prime to 3 prime direction, one codon at a time. So that is translocation. So translation and translocation together lead to elongation. Elongation happens due to combined effect of translation and translocation. Got the idea? But still, if you want to attach elongation to one of the two, then it is more associated with translation, because formation of the amino acid means elongating the amino acid chain. Formation of amino acid chain, sorry. So formation of amino acid chain means elongation of the amino acid chain is the elongation stage. Got the idea? Now termination stage. So the protein synthesis terminates when UAG codon, that is the stop codon, reaches A site. Now this is not the only stop codon. Actually it should be any of the three stop codons. Any of the three stop codons are reached. Means when any of the three stop codons reaches your A site, then protein synthesis stops, because stop codon does not have an amino acid associated with it. So that's why whichever tRNA comes and attaches, it does not have a amino acid on top of it, and that's why peptide bond cannot be formed. So then it gets released as it is, and there the protein synthesis stops. So this is a little out of syllabus, but I'm just mentioning that there are so many types of proteins, antibodies, contractile proteins. So antibodies are basically in your bloodstream. They fight with the viruses and external intruders, bacteria, etc. Contractile proteins, they are responsible for muscle contraction. So whenever you contract the muscle, it is due to contractile proteins. Enzymes are basically biocatalysts. So whichever chemical reactions take place in the body, they are catalyzed. That means their intensity is increased using enzymes. Hormones are the proteins which are messenger proteins. So for example, testosterone is a male sex hormone. So when this hormone is released by one organ, your testicles, then it travels through the bloodstream, it reaches all the parts of the body. When it reaches your body, it tells the hair to grow more. Okay, so testosterone is there, so that's why the hair will grow. If it reaches this part, it will say, okay, grow beard. If it reaches your muscles, it will say, okay, build muscles. Okay, so all these things happen due to testosterone. So what is testosterone? It is just a messenger. Testicles are sending this message across the body that, become a male. Okay, this is the message. Similarly, estrogen is a message that, become a lady. Okay, become a female. And so the breast enlargement will happen, and various other things will happen. Got the idea? So this is, all the hormones are basically what? Messenger proteins. Structural proteins are responsible for the structure of your body. Like that bones also have a protein called as collagen. Many people have brittle bones, and that is because they don't have enough collagen, right? So collagen is one example of this structural protein. It is also present in cells and in skin also. Even for cell integrity also, you require collagen. See, membrane has to be there, collagen has to be there for that. Any structure to keep things in place, you need collagen to some extent. Structural proteins are also found in every cell. Okay, and of course in the bigger tissues like bones, you require collagen. And but there are many other structural proteins. I'm giving just one example. Storage proteins, they reserve the amino acids in the body until ready for use. So ferritin is a storage protein which stores iron. So basically, whenever we have to store any nutrients, then that is also done using storage proteins. And then transport proteins. So they basically are the transport molecules, like hemoglobin in the blood. It transports oxygen from the lungs to all the parts of the body. Okay, so hemoglobin carries oxygen. What is hemoglobin? Hemoglobin is also a protein, and this kind of proteins which are used for transferring molecules from one place to another in the body are called as transport proteins. So these are the 7 types of proteins. This slide was put only so that I wanted to emphasize that enzymes, hormones, collagen, hemoglobin, these words you must have heard. All these are proteins, and that's why proteins are extremely important for the functioning of the body, and that's why this protein synthesis is important. So just to tell you the gravity of how important this situation is, this protein synthesis process is, that's why I told you where the proteins are being used in the body. These are all the types of proteins. Now think about antibodies. If we don't have antibodies, what will happen? Okay, so everything that is happening in the body is basically due to some protein or the other. You have a good height due to some protein. You have you have a male voice due to protein. Somebody told you, testosterone told this part, that uh, have a little relaxed laryngeals, and that's why a deeper voice can be generated. Everything is protein in our body. Okay, most of the things are by proteins. Carbohydrates, on the other hand, is only for energy. Mitochondria consume carbohydrates. Okay, fat is only for somewhere what is it called, to reduce the friction, or we need some fat so that we don't feel so much cold. So it's an insulator, it's a thermal insulator as well. So for that, fat is used. Got the idea? So I told you all these things, and then micronutrients are there, like vitamins and minerals are there. Oh, I'm not going into that that detail, but I told you the basic thing. Now, codons, how to use a codon chart. Codon chart comes in the form of a pie chart or in the form of a tabular kind of a chart. So how to use it? Suppose a codon is AUG. So the first letter you have to select here, A. Then second letter is U. So U. Okay, so A and U. So this is the square in which you have to check. And inside this you have to see, okay, what is the last one? G. So G. Okay. Okay. This means metanin. I think it is methylomin. Met means methylomin. So methylomin is the amino acid associated with AUG. So this is how you basically read a what is it called, codon chart. Okay, this is easy. Now you can understand that phi, or whatever that amino acid is, it is represented by two different codons. And this way, although we have 64 codons, there are only 20 amino acids. Let me still check once. 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20 21 22 23. Okay, there are 23 amino acids in human body, means which we use for protein synthesis. So 23 amino acids, and there are 64 codons. Okay, so that's how it is. I told you about one chart. The other chart is this pie chart. Personally speaking, I like the pie chart better because it's easier. Instead of searching here, I can go from here outside. So this is what? This is 5 prime and this is 3 prime. 5 prime, 3 prime, 5 prime, 3 prime, 5 prime, 3 prime. So you always move from 5 prime to 3 prime. Now let us say that the codon that I want to check is AUG. So here, where is A? A is here. Okay, I went here first. Then I will go in the next circle. In the next circle, there is again ACGU. So here I will go to U. So A, U. And then in the next circle, once again I will go to AUG. So this is what? This is the start codon, AUG. Methylomin. Methylomin is the amino acid associated with AUG. Got the idea? This also tells you that all the proteins in human body start with methionine. I don't know why is that the case. And there are these 3 stop codons. So these are stop codons. Here also stop codon, stop codon, stop codon. Okay. So these are stop codons. And this one is a start codon. So it is given in bold. Got the idea? So this is how you read a codon chart. This is only for the NEET students. This is not for 10th standard students, but you should know the entire picture. Whatever you write in the exam, of course, you can pressy write it, you can write it in short, but you should have the complete picture, right? And if you don't have the complete picture, it leads to a lot of confusion. That's why this science too seems to be a difficult topic for many students, because they don't get the whole picture. So I'm trying to give you the whole picture so that you can reduce it when you're writing the exam. You can reduce it. Okay. Now, what is central dogma? This will definitely not be asked in your exam, but this is for the NEET students. Central dogma of molecular biology is a theory stating that genetic information flows only in one direction. That means from DNA to RNA to protein, and not the other way around. Means based on the protein present in the cell, it won't form the DNA. That won't happen. Thank God, it doesn't happen like that. Okay, from DNA, RNA is formed, and from RNA, protein is formed. This is how the genetic information flows. That's okay, but it flows only in one direction. Again, sometimes RNA to protein. This is the case where you don't have a DNA, like in the case of viruses. But normally it is this trend, DNA to RNA to protein. So always the genetic information always flows in one direction, and this is called as the central dogma of molecular biology. 3.5 billion years ago, Earth was lifeless. Life did not exist on Earth 3.5 billion years ago. Then some simple elements existed in the ocean. Simple inorganic or organic compounds were formed from them. Complex compounds like proteins and nucleic acids were formed due to them. Nucleic acid means DNA and RNA. DNA and RNA came, that means life. So whenever DNA and RNA come, then life will come, right? So protoplasm, that is a basic cell, was formed. Okay. Unicellular organisms. So protoplasm was nothing but a unicellular organism. Okay. Now multicellular organisms. So they evolved into multicellular organisms. Darwin's theory of natural selection and Lamarck's theory of inheritance of acquired characters and the mutation theory, all these three tell us how evolution must have taken place. And all these three should be used together, because none of them is individually 100% correct. Where Darwin's theory lacks, Lamarck comes. Where Lamarck also lacks, mutation theory comes. Got the idea? So all these three happen together in nature. So all these three things have to be studied and considered together. They all these three things are the reasons for evolution. Okay, evolution happens due to all three of them. Gradual change occurring in living organisms over a long duration is called as evolution. So what is the definition of evolution? Few keywords. Gradual change, it's not a sudden change, occurring in living organisms over a long duration, that is called as a evolution. Okay, so change, like let us say if I go to the gym every day, I have built my body. That is not an evolutionary change. Will it pass on to my children? Probably not. But if he also does gym, or she also does gym, and their progeny also keeps doing gym all the time, suppose we are into a certain occupation which requires a great deal of physical labor, like an ironsmith. If I belong to an ironsmith's family, then of course my shoulders will be strong, because those strong shoulders will be inherited from my parents, and from my parents will inherit them from their parents, and so on. So from my ancestry, those strong shoulders will come to me. But it won't happen immediately. It won't happen because only I am doing gym. No, nothing. That is not a evolutionary change. That is your personal change. It's okay. Your one organism has changed, but that's not an evolutionary change. Evolutionary change means it happens over a long duration. Got it? So all these three keywords are important. Gradual change, it's a gradual change occurring in living organisms, okay, and over a long duration. Also, if you want to emphasize more, then you should be saying that these changes are such that they pass on to the next generation using natural reproduction. Okay, but this is the definition that your textbook has given, so I have just copied and pasted it over here. Biodiversity on Earth. At present, crores of species of plants and animals with huge diversity regarding shape and complexity are present on the Earth. Animal diversity ranges from unicellular and amoeba and paramoecium to man and giant whale. So paramoecium, you should be aware of what it is. It's a unicellular organism. Amoeba also, you should be aware of it. It was one of the first, or probably the first living organism, is called as amoeba. The plant diversity consists of various species ranging from unicellular chlorella. So chlorella is the unicellular plant, and amoeba is a unicellular animal. Paramoecium is a unicellular animal. Okay, these things are more for the NEET students. less for the 10th standard students, but still you should be aware. Amoeba, Paramoecium, these are animals. Chlorella is a plant, and all these three are unicellular, having single cell. Okay, so plants also have a lot of diversity, starting from unicellular chlorella to huge banyan trees. The life exists on Earth everywhere, from equator to the poles. Actually, the life seems to be wrong, but this came directly from your textbook. There are many grammatical mistakes in your textbook. Everywhere, wherever they have said hair, it should be hair. They have said hairs. Hairs is a wrong word, but I don't know why. Even in the textbook, these mistakes are there. So here also, I think the should not be in front of life. Life exists on Earth everywhere, from equator to both the poles. Organisms are present in all the places like air, water, land, rock, etc. Evidences of evolution. So this can be a good question. There are 7 evidences of evolution. I'll explain them one by one. Morphological evidences. If the external appearance is the same, then those organisms must be derived from a common ancestor. Like dogs, cats, wolves, all of them have similar snout, similar fur, similar limbs, similar ears, similar eyes. So we can assume that initially their ancestor was the same, and then they gradually evolved into different types of organisms based on why did this happen? This happened because they reproduced in separate pools, means they were separated reproductively. A cat and a dog don't reproduce together. That's why dogs will only reproduce amongst dogs, and they will keep on growing their features in a different way. Cats will only reproduce among cats, and that's why they will keep growing in their own particular way. Okay, so since there is reproductional isolation, these species developed into different species. Means these one, maybe it could be a single ancestor, but that ancestor then that one ancestral species got evolved into 2 different species, dogs and cats. So based on physical appearance, that is morphological appearance, we are trying to justify that this evolution must be true. Why? Because we see morphological similarity, that is external similarity. Morphology means external appearance of an organism. Anatomy means internal structure of an organism. Now see, sometimes externally they might look different, like a human arm, cat's arm, bat's wing, or whale's fin. They all look different. But if you see their anatomy, if you see the bone structure, then you will find that the number of bones is the same. Bones could be long or short, that's another thing, but basically the bones are the same. Like in bat wing, the fingers are really long, and that basically forms most of the bat wing. But overall, it also has fingers. We also have fingers. Then the joints amongst these bones is also similar. That means the anatomy of humans, human hand and cat arm and bat wing and whale fin, they are all anatomically same. Internal structure is the same, and this also suggests that probably all of them came from a common ancestor. And this again says that one thing can evolve into many things. That means theory of evolution is again proven by anatomical evidences. Then vestigial evidences. Degenerative, that is underdeveloped organs, are called as vestigial. If an organism has a vestigial organ and other organism has the same organ in a well-developed form, then it means both of them must be having common ancestry. For example, appendix is vestigial in human beings, but appendix is functional in ruminants. Who are ruminants? Ruminants means cows and buffaloes. जी जनावर रवंत करतात त्यांना रुमिनंट्स असं म्हणतात. रवंत करणं म्हणजे काय? मीन्स what is ruminescence? Basically they eat food immediately because they have to graze on the pastures while keeping themselves safe from the wolves and maybe not wolves but tigers, lions. So they have to graze quickly, and then when they are sitting in a safe place, at that time they bring back that food from their stomach to their mouth and they keep chewing it all day long. This is called as ruminants. याला रवंत करणे असं म्हणतात. कळले काय? So that is ruminant animals. So ruminant animals are cows and buffaloes. Okay, so they have well developed appendix, because appendix is required for digestion, because they are eating raw things. We don't eat raw grass, right? But cows and buffaloes eat it. That's why they require appendix. So then probably both of us have appendix. What is the use of appendix in my body? Nothing. Still I have it. Why? Because probably both of us, both the species came from a common ancestor. Similarly, movable pinna. It is vestigial in humans, but it is well developed in cats and dogs. See, pinna, we don't even require it. We don't need this thing. This thing hanging around, we don't need it, except for hanging our spectacle. We really don't need it. But cats, dogs, wolves, they require it. Wild animals do need it because it can collect sound from a certain direction. Now, this one is always collecting sound from this direction only. But imagine if I could deviate it like this and bring it forward, so it would pick the sound from front. Suppose I can move it backwards, so it can pick the sound from backwards. It is particularly good in a jungle situation. I don't live in a jungle, I don't require that. Humans don't require it, and still we have pinna. Why do we have a pinna? Because humans, dogs, cats, wolves, all of them have come from a common ancestry, and that's why some of the parts are vestigial in us, because we have evolved in and we have changed the situation around us, so we don't require pinna so much. But yeah, some other species do require it, and all of these species have come from a common ancestor. So theory of evolution is proven again. Body hair, coccyx, that is tailbone, wisdom teeth, these are all vestigial too, but they might be important in other species. Like body hair is required in chimpanzees, gorillas, because they live in the wild. Wisdom teeth, I don't know who uses it or who needs it, but tailbone, people, some species have tails. Okay, monkeys have tails. Apes don't have tails, but apes still have the tailbone, and from apes we came. Humans also have tailbone. That means humans came from apes came from monkeys, all of us came from common ancestry. That means theory of evolution is again proven. So this is vestigial evidence. Next evidence is paleontological evidence. Paleontology is the study of extinct species. It uses carbon dating to identify how old a fossil is. What is a fossil? Fossil means, let us say there were dinosaurs. Let us say means it's a reality actually. Dinosaurs were there on planet Earth. So then meteor hit the Earth, and due to this there was cold age, ice age, and they all died into it. Now what happened is, when they died, then the dinosaur is dead, the flesh will rot away, or maybe it will be eaten up by other organisms, but the bone will still exist. Now who eats bones? Yeah, no one eats bones, right? Bones exist. Then what will happen? Let us say there is a landslide, it is covered by mitti, earth, soil. Okay, suppose there is an earthquake, it fell into the soil or earth. Suppose there is a volcano, so those sedimentary rocks, sorry, not sedimentary, they're igneous rocks were deposited on top of this skeleton. So that skeleton is now preserved. Now no one is able to disturb it. It is preserved. Who will dig it out and destroy it? No one will do that, right? So it is very much intact. So whenever we excavate, that means we dig into the earth to find certain things. So basically we are trying to find skeletons of dead organisms so that we can study more about them. Or excavation can also be done to excavate some historical site like Mohenjo-daro, Harappa, there. So excavation basically means digging the earth to find something valuable. So here in biological terms, we'll say that we are trying to find skeleton of dead organisms. Now, there are various layers of earth. So in the upper layer, these must be latest organisms which died recently. Below them, there must be organisms which must be dead way beyond those organisms. So this way, the lower you go into the earth, you will find the fossils of older organisms. Okay, so fossils means the remains of prehistoric organisms. They are called as fossils. Now, one way of telling how old that fossil is based on how deep that fossil was found. Okay, it gives you a rough idea that it is found so deep into the earth. That means it must be dead since a long while. So it must be a very, very prehistoric organism. Okay, other than that, what? How will you precisely say that exactly how many million years ago this thing existed or walked on the planet Earth? For that, we use carbon dating. So here are the steps for carbon dating. Carbon 12 and carbon 14 are isotopes of carbon. Okay, isotopes means same atom, same molecule existing in two different forms. So those two different forms are called as isotopes. So the heavier isotope is mostly, many a times it is radioactive, because see, if there is see, basically what happens is whenever there is a heavy isotope, it will degenerate, okay, to create a lighter thing. Okay, so basically if you throw a ball from this height to the ground, it will try to come from high potential to low potential, right? So this isotopes also, usually heavy isotopes are radioactive, so they release their energy and they get converted into a little lighter thing, and this release of energy is in the form of light, and that's why they are called as radioactive. What the idea? This is radioactivity. So carbon has two isotopes, lighter and heavier. Lighter is C12, heavier is carbon 14 or C14. So this heavier isotope is radioactive. Okay, now they exist in the nature in certain proportion, and that proportion hasn't changed since millions of years. So just think about it, that suppose, I'm just giving hypothetical thing, that ratio of C12 to C14 in nature is X is to Y. Okay, must be 5 is to 6, 17 is to 7, anything. I don't know what is X and Y, but I'm saying there should be some ratio of C12 is to C14. And that ratio has not changed for millions of years. Okay, now what will happen? C12 is radioactive. Okay, both of them form carbon dioxide. Carbon dioxide is used by the plants for photosynthesis. Plants are consumed by animals. Animals are consumed by other animals. So basically, this carbon in both these forms enters every human being's or every organism's body. Okay, so it must be the part of the body of dinosaurs also, right? Dinosaurs must be eating plants. Dinosaurs, carnivorous dinosaurs must be eating other dinosaurs or other organisms at that time, right? So this carbon reached into their body, and this proportion X is to Y must be there when they were alive. But when they are dead, so as long as they are alive, this proportion stays constant, and this proportion is this, that C12 is 1.35 into 10 raised to power minus 12, and C4 is only 1. So this ratio is often given as C4 is to C12. Why? Because here 10 raised to power minus 12 is given. So if it is in the denominator, then the value will be positive. That's the reason why they give C14 is to C12. Here in the entire slide, I have said C12 is to C14 because I thought 12 comes first, 14 comes later, that's why. But normally, if you see any material, they'll say C14 is to C12 ratio. Okay, and so when you say C14 is to C12 ratio, then just revert this. You will say 1 is to 1.35 into 10 raised to power minus 12. Got the idea? Now, when the animals die, now they are not eating anything, so carbon proportion, carbon won't be included into their body. New carbon is not coming into their body. Now this old carbon is C12 and C14. C12 will stay as it is because it is non-radioactive, but C14 will degenerate into lighter things. So C14 will reduce over the time. And radioactivity means, see, radioactive means I am also radioactive in that way. I also have C14. Do you see any radioactivity in my hand? You don't see, na, because that radioactivity is very, very slow. So they degenerate, it degenerates, but it degenerates very, very slowly. So it will take almost infinite amount of time to completely consume this C14. Means this C14 for getting degenerated and getting vanished from the body, it will take almost infinite amount of years. And this is very important, because now what we'll do is, whatever fossil we got, we will see what is the proportion of carbon 12 and carbon 14 into it, and based on how much carbon 12 has degenerated, we can determine how old that fossil is. And this is called as carbon dating technique, because we are dating a fossil, that how old this fossil is, based on the carbon content inside it. So it is used in paleontology, which is the science of ancient life, as well as anthropology, which is science of humanity. Even if you find human fossils like Neanderthal, so how do you know that Neanderthal was these many years old? Because we did carbon dating of the skeleton of Neanderthals. And that's why we know that they are so and so years old. So they walked on the earth so and so years old ago. Carbon dating indicates that vertebrates evolved from invertebrates. So paleontological evidence shows us that, most of the skeletons that you see, they are mostly like our skeleton. So previously, they did not have vertebrae. Now they started having vertebrae. So that means having a vertebra, that is having a spinal column, not spinal column per se, but this bone structure on the dorsal side of the body, that is called as vertebra. So having a vertebra is an evolution. Why? Because older ones did not have vertebrae, and the newer ones have vertebrae. That means vertebrates must have evolved from invertebrates. Okay, so paleontology also gives us an evidence of evolution. It tells us that evolution is true. Another evidence is connecting links. So some plants and animals show some morphological characteristics which connect them to two different species. For example, platypus is a connecting link between earthworm and cockroach. So like earthworm, it has segmented body, thin cuticle. Cuticle means skin. Parapodia like organs. Parapodia means hair like organs for moving. Hair like organs are called as parapodia. So just like an earthworm, peripatus has these all characteristics. But peripatus also has certain characteristics like cockroach, like tracheal respiration. Tracheal respiration means cockroaches don't have lungs. They breathe the air within the body and exhale it, and that means the entire trachea is responsible for respiration. They don't have a specific organ. Okay, trachea can also be considered as an organ for respiration, but they don't have lungs, in short. So that is tracheal respiration. And then circulatory system, they don't have a closed circulation system, they have an open circulation system. Blood is not circulated using veins and arteries. It does not have blood vessels to carry the blood. All the blood is in the pouch form inside that cockroach. So that this is called as open circulation system. So open circulation system is found in cockroaches. It is also found in peripatus. So that means peripatus must be a connecting link between earthworm and your cockroach. Earthworm must have evolved into peripatus. Peripatus must have evolved into cockroach. Similarly, duck-bill platypus is a connecting link between reptiles and mammals. So it lays eggs like reptiles and it has mammary glands and hair like mammals. Reptiles don't have hair, you know that, right? Mammals have hair. And even those hairs were more in monkeys. In apes, the face was not having hair. And in humans, most of our body does not have hair. Very few hair are remaining, right? So because we do not require an exoskeleton, we don't require so much protection from different temperatures, because now we can wear clothes, we can protect our body otherwise. So we don't actually require hair. Okay, lungfish can be a connecting link between fish and amphibians. So lungfish looks like a fish, but it can also breathe outside the water because it has lungs. Amphibians basically always live outside the water, but they live near the water. Okay, like frogs. You will never see frog in, means of course they can swim inside the water, that's all possible, but most of the time they are on a leaf or on a rock, but that leaf or rock is always very close to the water. So they will keep going into the water, coming out, going into water, coming out. एक दो दिल एक जान टाइप रहते हैं पानी और amphibians. Crocodile also is an amphibian. You will never find a crocodile, okay, chilling out somewhere in a desert. No, you don't find them like that. You find them near oasis, you find them near water. दो दिल एक जान होते हैं ये लोग, ठीक है. They cannot separate from each other. So these amphibians must have evolved from fish, and the connecting link between the fish and the amphibian must be this lungfish. Embryological evidences. So initial stages of embryos of all the species are very similar. This indicates that all the species evolved from a common ancestry. Darwin's theory of natural selection, also called as survival of the fittest. For this purpose, Darwin published a book titled Origin of Species. In Origin of Species book, he mentioned the theory of natural selection, also called as survival of the fittest. All the organisms produce prolifically. Prolifically means too much. Like if you see cockroaches, they give birth to a lot of babies. Okay, many of them die, but then the species still survives. Why? Because in spite of all those hardships, some of them are able to survive so that their progeny continues. Rabbits, they give birth to a lot of offsprings. Why? Because many of them are eaten up by wolves and all the other wild animals, but some of them still survive because the rate of reproduction is more. So that is prolific reproduction. All the organisms compete with each other in life-threatening manner for food, survival, mating. Okay, survival of the fittest means only the fittest survives. Only those organisms live which show modifications essential for survival. So maybe the person with strong and stout body will survive. He will mate. Even a woman with suppose polio, she won't be able to reproduce because people won't like to have mating with her, and they will mate more who are good looking, who are strong and able bodied. They will reproduce more. That's why survival chances are more for those people who are best fitted for survival, who are having able body. They will survive better. Sustaining and selected organisms can perform reproduction and thereby give rise to the new species with their own specific characters. Okay, so of course this is what I have already explained. Darwin's theory of natural selection was widely accepted for a long duration. Objections to Darwin's theory. Now these objections came later on. Previously, for a very long time, he was accepted. Everyone accepted. Very good. For a long time, his theory of natural selection was accepted. However, some objections were raised against his theory. So what are those objections? Some of them were raised during his lifetime only. Okay, so that also happened. I'll tell you what they are. So these are the objections which your textbook gives. Okay, and the in gray whatever I have given from my own mind. Okay, so I did not understand what they wanted to say. Arey, कहना क्या चाहते हो? That's how they write their textbooks, unfortunately. So I tried to explain it in those gray letters. I tried to explain. I tried to understand what क्या कहना चाहता है भाई. I tried to understand it. So natural selection is not the only factor responsible for evolution. This was first objection. That means there must be some other factors also. What are these factors? So few modifications happen due to mutation also. So that is another reason for evolution. Okay, I told you earlier that theory of natural selection, then Lamarck's theory of use and disuse of organs, and mutation. All these three are responsible for evolution. So natural selection is not the only factor responsible for evolution. Second thing, Darwin did not mention any explanation about useful and useless modifications. So If everything was meant for the survival of the fittest and everything, then only those modifications should pass on which are actually adding value or survival value to that organism. But then some modifications are absolutely useless. For example, everyone has different fingerprints. Absolutely useless. Handprint, absolutely useless. Some people have joined earlobes. Some people have separated earlobes. Absolutely useless modification. What is the use of it? So why did these modifications also pass on? That Darwin does not explain in his theory of natural selection. Then there is no explanation about slow changes and abrupt changes. Slow changes means what? Example of a slow change is degeneration of vestigial organs. So our pinky finger is reducing in size. Our wisdom teeth, normally people don't even have wisdom. I mean, that wisdom teeth does not grow out of the gum. So this is an evolutionary change. These organs are becoming vestigial. Appendix, it never grows to the full size. So if it grows, then we have to surgically remove it. So that is basically what these organs are becoming vestigial. Our tailbone is becoming vestigial. So these are slow changes. Why are changes so slow? If you don't require a pinky finger, why do you still have it? What's the use? Can't there be some human beings who won't be having pinky finger? They'll be more suited for life, and then they will reproduce more. So this theory of natural selection does not give an answer to it. This is what I felt. Quite frankly speaking, now these 3 lines, I did not understand what the textbook writer wanted to say. कहना क्या चाहते हो? I could really not understand. But this is the best that I could comprehend out of it. You let me know in the comment section if I'm wrong and if you know something better. Example of abrupt change. Wolves find a way to survive in the radiation in the Chernobyl exclusion zone. What is Chernobyl disaster? Ukraine had nuclear weapons earlier when it was part of USSR. At that time, there was the nuclear facility. There was an accident at the nuclear facility and there was a radiation. That is called as Chernobyl disaster. So of course, just like humans, animals were also affected due to radiation. But certain wolves found out a way to live in that radiation. How did they survive? That was an abrupt change in them. How did they, and see, this change was observed in many wolves. That means the entire species, it was an entirely different species of wolves who could survive in a radiation environment. Such a huge change happened in one generation. We say that evolution is a gradual process, but such a huge change happened in one generation only. How is it possible? This is not explained by Darwin's theory of natural selection. Now, principle of use and disuse, that is Lamarckism. It was given by a French person, Jean-Baptiste Lamarck. Lamarck proposed that morphological changes occurring in living organisms are responsible for evolution. Okay. The reason behind this morphological means external appearance. Morphological changes is activities and laziness of that organism. Means that or that part of the body will grow better which you are using. So the more you use something, the more well developed it will be in your offsprings. But the less you use a certain part of the body, the less the less well developed it will be in your progeny as well. So over the years, this finger is reducing in size. The load carrying capacity is reducing, power is reducing. Why? Because we don't use this finger. Especially toes, we are not using toes, and that's why see, at least the hand fingers are these long. Toe fingers are so small because we don't even use them. So laziness, if you don't use something, then it will cease to exist in the upcoming generations. Okay. And if you use something more and more, like giraffe wanted to eat leaves higher up the tree. So those giraffes could reproduce better who had longer neck, and those who had shorter neck, they did not get any food because longer neck wala could eat the lower leaves also and higher leaves also. So longer neck giraffe was at advantage. So they reproduced more, and the shorter ones died, so they could not reproduce. So now giraffe has so much longer neck. So if you use neck more, your neck will improve. If you use something less, then that something will reduce. Okay, so this is basically the long and short of Lamarckism, that is principle of use or disuse. Examples, I've given so many examples. Giraffe, what else? Ironsmith. Yeah, if you're into a family of ironsmiths, then you may be having stronger shoulders. Wings of birds like ostrich, emu, have become weak because they never used it. Chickens also, they don't fly. Ostrich doesn't fly. Emu doesn't fly. So their wings are not very strong, and they have smaller wings also, by the way. But take the example of bat. Arre baap re, most of the span is wingspan only because it flies so often. Then legs of the birds like swan, ducks have become useful for swimming because they have webs. So their legs have developed webs so that they could swim. So they swam so often that, those having an abnormality, let us say that there was a web, they had a better chance of survival. So they had better chance of reproduction also. So more and more that kind of people, that kind of swans started existing. And then those swans who did not have the web, they were at a disadvantage. They could not reproduce so often. Okay, so their genetic signature reduced, you can say. So now all the swans, all the ducks, they have webs. Okay, snakes lost their legs by modifications in their body for burrowing habit. Snakes were in a habit of burrowing. So now when you get inside a burrow, that means burrow means a hole dug inside the earth. So if you live in a burrow, you don't require long hands, otherwise they'll come in the way, right? So snakes did not require arms, they did not use them, so they fell off. Fell off means they reduced in the size, and one fine day probably snakes were born without hands. So this is how they didn't use it, that's why they were lost. Why Lamarckism was disapproved? Now, just like Charles Darwin, Lamarck was also not fully accepted. Development of organs due to specific activities or degeneration due to in use was widely accepted. This much is accepted. But transfer of those characters from one generation to another was rejected. Okay, because it had been verified many times that modifications brought in us are not transferred to the next generation, and thereby Lamarck's theory was disapproved. See, what people said is, okay, you say that, blacksmiths, yes, okay, I have built up my body. It doesn't mean my child will also have good body. The child has to build his or her body on their own. Okay, so this is an objection to Lamarck's theory. People say that you haven't specified how exactly is the transfer of modifications happening. Just like Charles Darwin did not say that how modifications happened, you did not say how these modifications are getting moved or carried forward across the progeny. That you haven't said, and that's why this is partially accepted, partially rejected. What is mutation? Mutation theory says that a mutation is a change in the genetic sequence of an organism. So for mutation, see the movie X-Men. All those are mutants. Or mutant ninja mutant. There are some turtles who are mutants. So mutants can result from errors in the DNA replication during the cell division. So whenever cell division happens, DNA gets packed up into chromosomes and then they divide and whatever happens. Okay, so that is called a cell reproduction. During this, let us say that certain gene was not properly copied. So every chromosome has to be copied, right? From one chromosome you need two chromosomes now. Maybe during this copying thing, some codogen could not be copied properly. Okay, so that can lead to mutation. Okay, so the amino acid for that codon would be different, so the protein formed would be different, so it will have some different bodily function, and this is called as mutation. Now, germline mutation, it is called as germline. Germline mutation means that occur in eggs and sperm. They are called as germline mutations. So germline mutations can be passed on to the offsprings, while somatic mutations means which happen in large organisms, means fully grown organism. So in me, if a mutation happens, then it is somatic mutation. But in suppose when I was in the zygote state, I was one cell at that time, that my first cell is called as zygote. If some mutation happens at the zygote state, then it will be passed on to all the because all the cells in the body will be created as per that mutant copy. So all the cells will have that mutant gene, and that's why that entire human being will have mutant genes. So the sperms created by that main man will also have mutant genes, and they will you know combine with the egg of the female. So that mutant gene will come here. Now suppose her gene is normal genes are usually stronger than mutant genes, so if her gene is able to mask this mutant gene, then it is okay. But if it is not able to mask it, then the mutation will carry on in the child. So for mutation to happen, two things are needed. One is the mutation should be germline. Then and only then all the cells in your body will have that mutation. Then the sperms formed by you and the eggs formed by the lady will have that mutation. Second thing is both of you should be having the mutation. Then and only then, for a certain trait, if both the genes are mutant genes, then both are weak. If both are there, then they can do something. But if one of them is strong, then the strong one, natural gene will always be, normal gene will always be masking the effect of mutant gene. So there are two things required. One is it should be a germline mutation. Second is the germline mutation should be in both the organisms. It should be in the male as well as the female. Then only it happens. Otherwise, usually mutant genes are weak genes. Usually the mutant gene is weak. It gets masked by the chromosomes. It gets masked by the normal gene because chromosomes are always in pair. For example, mutation may cause genetic disorders like sickle cell anemia. Yeah, this is an example in your textbook, so remember it. Sickle cell anemia is caused due to mutation. Speciation. What is speciation? Speciation is formation of new species. Species are a group of organisms that produce fertile individuals through natural reproduction. Okay. Reasons of speciation are mutation, that is genetic variation, geographical isolation leading to reproductive isolation. So if one species is not able to reproduce in a common pool, one pool is reproducing separately, another pool is reproducing separately, then they will develop separately. They will evolve into two different species over the time. So why does this happen? Due to geographical isolation many a times. Like there could be different islands on which, tiger used to be there on a certain island, then water table rose, or sorry, water level rose. So that area was divided into multiple islands. So now tigers on different islands will grow differently. They will have minute differences, and then those differences will grow on over the time if the geographical isolation stays like that. Okay, so geographical isolation, which leads to reproductive isolation, will lead into speciation. Species will get formed. Human evolution. 7 crore years ago, dinosaurs disappeared. Lemurs evolved into monkeys. 4 crore years ago, in Africa, monkeys evolved into apes. Different differences between monkeys and apes. So monkeys have tails, apes don't have. Apes have larger and wider chest. Apes have their faces without hair. Apes are usually stronger than monkeys. So apes, that's why you have Planet of Apes. Why? Because these are stronger versions of monkeys, and we are the intelligent version of apes. And mind is mightier than sword, and that's why we rule the planet now. Apes reached south and north of Asia. So until now, the ancestors of humans were in Africa only, but now they came to Asia as well, south and northeast of Asia. They evolved into gibbons and orangutans. Then the apes which stayed in Africa evolved into gorilla and chimpanzees. Okay, and here in Asia, gibbons and orangutans. 2 crore years ago in Africa, they started to stand erect on the grasslands. So in the grasslands, they tried to stand erect. They were still not very erect, but they could at least stand so that they could see more area. They could see if someone is attacking you. They could see if there is any prey that I can attack. For that, they could stand. Although not very upright, but they could at least stand. That is called as Dryopithecus. Okay. Now after that, 1 crore years ago, first human-like animal in Africa and North India was found. It is Ramapithecus. 40 lakh years ago, apes grew in size and became wiser. Ape of South Africa, also called as Australopithecus. 20 lakh years ago, skilled humans, that is the first humans who could make weapons and other tools with stone and wood. So Stone Age started, you can say. So first human, and that's why they are called as Homo. Homo means human. So Homo habilis. Till now we have reached Homo habilis. After that, Homo erectus came. 15 lakh years ago, humans walking in erect posture came. They were called as Homo erectus. Their fossils were found in China and Indonesia, so definitely they must be in China and Indonesia. After that, 1.5 lakh years ago, first wise human who lived in caves, used fire, and lived in tribes. And this human was called as Neanderthal. 50,000 years ago, Cro-Magnon came. So Cro-Magnon is a little more evolved as compared to Neanderthals. Rate of evolution increased after this stage. After Cro-Magnon, the rate of evolution increased a lot. Evolution of human beings increased a lot. 10,000 years ago, we did agriculture, reared cattle. Reared cattle means rear याने palna, याने भेड़ पालन, पशु पालन. That is called as rearing cattle. And established cities. Culture developed. See, agriculture is also a culture of growing crops. So then few cultures developed. Like there would be weddings. There would be, I don't know whether that culture, but I feel that by culture they mean these kind of things, that living in a tribe, so there should be a mukhya of the tribe, the leader of the tribe. Then there should be some rituals. If someone dies, then there will be some rituals, whether you will bury them or you will float them in a river or you will burn them. What are the rituals that you will do at that time? This means culture. Whenever any rituals come into play, then it is culture. Whenever agriculture comes into play, it is culture. Whenever any kind of civilization is there, it means culture. So the moment you started living in tribes, culture started forming. So culture actually started forming here itself. But by culture developed over here, probably they are meaning to say that they had some rituals also now. 5,000 years ago, art of writing was invented. 400 years ago, modern science emerged, and 200 years ago, industrialization started. So during British Raj, industrialization started. Human evolution. So this is taken from your textbook. So these are the years, and so that you can remember very easily. Okay. Now there is one question that I saw in the exercise part of your textbook, and it says there is evidence of fatal science among chordates. Again, कहना क्या चाहते हो यार? यानी textbook में भी whatever you write in the textbook, it is also so garbled. It's difficult to understand what you want to say. How can you justify a statement that you can't even understand? There is evidence, okay, of fatal science. What is a fatal science? Among chordates. First of all, you never explained what are chordates throughout your chapter. I had to Google it and find it, who are chordates. So this is my explanation. If this question comes to you, then probably this is the answer that you should be giving. Of course, I picked it from a website, but this is what was given on the website. It is believed that chordates have originated from invertebrates. So chordates are organisms which originated from invertebrates. So previously, all the organisms were invertebrates. None of them had the vertebra column. Okay, vertebral column. Chordates were the very first organisms who built something like vertebral column. Okay, and that is why this is first point. It is difficult to find out the invertebrate group from the chordates from which the chordates were developed. So you could not find their ancestors, chordates ancestors. You could not find. The ancestors of chordates are believed to be soft-bodied animals, and hence they were not preserved as fossils. So probably the invertebrates who were the ancestors of these chordates, they were so soft-bodied that they did not have any bones in their body, or at least they didn't have a structure like us, because our entire bone structure hangs by our vertebral column, right? So if that vertebrae itself were not there, even if bones might be there, they could not function and they could not move and that kind of a thing. Or probably they didn't have bones itself, and that's why their fossils could never be found. Thus, the evidence of fatal signs among chordates. So that means, since you could not find the ancestors of chordates, that means their ancestors must be those who didn't have bones. So if they didn't have bones, they didn't have the vertebral column as well, and that's why their ancestors were invertebrates. And this is how they tried to prove that chordates, who were amongst the first vertebrates, they originated from invertebrates. Okay, so fatal signs, by fatal signs probably they meant to say that you cannot find the ancestors of chordates, and that's why see, fatal signs probably means the study of fossils, something like that. So you could not find the fossils. You found the fossils of chordates, you could not find the fossils of their ancestors. So that is fatal signs, study of both these fossils. One was existent, another was not, and this proves that chordates originated from invertebrates. I think that is what this line says, but really, let me know in the comment section what it means. How do they make textbooks here? I mean, this is a line in the textbook. What is, I do not understand it at all. How can I justify it here? You first write it in proper words, then I can explain it. And what are chordates? I had to search it online. You didn't even mention it in your textbook. Okay, some scientists' names. I don't think they'll be asked, but you should know. So I'll just read out their contributions. This is taken exactly from your textbook. Sir, I think he was Sir only. Sir Gregor Johann Mendel is pioneer of modern genetics. You will study this guy in 11th and 12th also, and it will be needed for your, this thing, pre-medical test or NEET students. You will need it. So Mendel is the guy who researched over pea pods or pea trees, and he tried to study their genetic their genetics. So he is the pioneer of modern genetics. It took a long time to understand the conclusions of his research about heredity. In 1901, means it is very recent. How many years ago? 124 years ago or 123 years ago. The reasons behind the sudden changes were understood due to the mutation theory of Hugo de Vries. This can be a question. Who is Hugo de Vries, or mutation theory was given by whom? Hugo de Vries. Meanwhile, just the next year, Walter Sutton observed a pair of chromosomes in the cell of grasshopper. Until then, it was not known to anyone. So chromosomes were discovered for the very first time by Walter Saturn in 1902, means almost 120 years ago. Research started in the direction of finding the nature of genetic material, and then when it was proved that genes are carried via chromosomes, through which 1944 trio of scientists, means three scientists, Oswald Avery, Macklin McCarthy, Colin MacLeod, they proved that except viruses, all living organisms have DNA as the genetic material. So this DNA, discovery of DNA, or at least considering that DNA is basically genetic material, this happened in 1944. 1944 is like, what can you say, how many years ago? Today is 2024. So 60 and 20 means almost 80 years ago. This 80 years ago we came to know about DNA and what is the significance of DNA. In 1961, the French geneticist Francois Jacob and Jacques Monod proposed a model for process of protein synthesis with the help of DNA in bacterial cells. Okay, so 1961. Then this is an Indian guy, Dr. Har Gobind Khurana. Har Gobind Khurana ji, a scientist of Indian origin. That means he was not an Indian. He probably took the citizenship of some other country, must be the US. Has made an important contribution in the discovery of triplet codons. So you have heard about codons all through this chapter. So codons, a significant amount of discovery about codons was done by Hargobind Khurana ji along with two other scientists. Okay, so a discovery of triplet codons for 20 amino acids. So out of how many? 23 amino acids are there in the body. 20 amino acids, the codons were discovered by these trio, Hargobind Khurana sir and two more scientists. Okay, so we should be proud about this Indian guy. And of course, he was awarded a Nobel Prize in 1968 along with two other scientists. Charles Robert Darwin, you know about him, gave theory of natural selection. And Jean-Baptiste Lamarck. Lamarck is not M-A-R-K, it is M-A-R-C-K. So the pronunciation should be Lamarck. Lamarck gave theory of inheritance of acquired characteristics, also called as principle of use or disuse of organs. Actually, this is the correct name, principle of use or disuse of organs. This is also given in your textbook. This acquired characteristics, inheritance of acquired characteristics, I have taken from one of the YouTube videos. Okay, so don't take this very seriously, but this given in the bracket is the one that is given in your textbook. That is principle of use or disuse of organs. That is correct. Okay, and these are all the sources. Again, important sources, other sources, all of them have made this video possible. So thanks a lot to all of them. These are important, means you must go through them. This is your textbook, so you should go through your textbook. And these two are some YouTube videos. One YouTube video, I took the snapshot of that and I put it in this slide itself, and another YouTube video was of another lecturer just like me, and it was also great. So do go through all of them if you have time. Until next time, Jai Shri Ram. ये है अमित शिरासाव, अमित शिरासाव, अमित शिरासाव, अमित शिरासाव.